Hello everyone. In the last few videos, we learned about the basic structures of macromolecules in the living system. DNA and RNA are the two types of nucleic acids in living systems, named so because they are acidic and are found in the nucleus of the cell. It was Rosalind Franklin who first experimentally obtained the X-ray diffraction images of DNA, which led to the discovery of the structure of DNA, a double helix, for which James Watson, Francis Crick, and Maurice Wilkins shared the Nobel Prize in Physiology or Medicine in 1962. DNA acts as genetic material in most organisms. That is, it is the molecule that is stably passed down from cell to cell and from generation after generation, and it is the cause of heredity. RNA, on the other hand, acts as a genetic material in some retroviruses, but mostly as messenger, adapter, and structural molecule. In this video, we are going to discuss the process of making more DNA from DNA, that is replication, and making RNA from DNA, that is transcription. In every cycle of replication, the amount of DNA present doubles, such that there are two identical copies of the initial DNA. The way it happens is that the two parental strands of DNA unwind, and each of them acts as a template, a sort of photonegative or a stencil on which the new complementary strand is made. Complementarity means that A always binds to T and G always to C, like how puzzle pieces fit into each other like a lock and key pair. This way, if you know the sequence of one strand, you know the sequence of the other one too. So, after one cycle, both the DNA molecules will have one parental strand and one newly made strand. This is known as semi-conservative replication. After another cycle, you have 4 molecules, then 8, then 16, and so on. In just 20 cycles, one molecule can end up forming more than 10 lakh identical molecules. A human body is made up of around 30 to 40 trillion cells, and each cell has the same DNA. When we consider the fact that each individual starts from one cell and that new cells replace old cells all the time, even while you're listening to this, you can imagine how important the making of DNA is. Now, let's move on to transcription. The process of copying genetic information from one strand of the DNA into RNA is called transcription. Here also, the principle of complementarity, that is AT and GC, takes care of the process of transcription, except that A now forms the base pair with U, that is uracil, instead of T, thymine. During replication, the complete DNA of an organism gets duplicated or doubled. But in transcription, only the required part of DNA and only one of the strands is copied into RNA. This means that the DNA has to be marked to indicate which part of which strand and what length of it has to be transcribed or made into RNA. It is done in the following way. Each segment of DNA that forms an RNA molecule is called a transcriptional unit, which is defined primarily by three markings: a promoter or the start signal, the structural gene or the part that is actually copied, and a terminator or the stop signal. The process goes like this. An enzyme comes and binds to the promoter and starts to transcribe. That is, it starts to arrange the nucleotides AUGC according to complementary sequence of the nucleotides TACG on the structural part of the DNA strands. When the enzyme reaches a stop signal, it breaks off and the RNA molecule is released. In the next few videos, we will see how this mRNA molecule released is translated to protein and learn about the genetic code. Until then, goodbye.